Hello friends, I am Tehseen Khan, Assistant Professor at Bhagyodit Pharmacy College, Sagar. Friends, we were discussed about drug metabolism. In the continuation of drug metabolism, today we will deal with various oxidation aspects of drug metabolism, including oxidation of aliphatic and alicyclic carbon atoms, oxidation of carbon-nitrogen systems, and oxidation of carbon-sulfur, which is scripted by Professor R.K. Agrawal, Dean of Science and Technology, Dr. Hari Singh Gaur University, Sagar. The metabolism of drug and other xenobiotics is often a biphasic process in which the compound may first undergo a functionalization reaction, that is, phase 1 reaction of oxidation, reduction or hydrolysis. This introduces or unveils a functional group such as a hydroxyl or amino group, suitable for coupling with an endogenous molecule or moiety in a second metabolic step known as conjugation reaction, that is phase 2 reaction. In a number of cases, phase 1 metabolites may be excreted prior to conjugation, while many xenobiotics can be directly conjugated. Furthermore, reactions of functionalization may follow some reactions of conjugation. For example, some conjugates are hydrolyzed and or oxidized prior to their excretion. Oxidative biotransformation processes are by far the most common and most important in drug metabolism. The enzyme systems carrying out the biotransformation are referred to as mixed function oxidases or monooxygenases. There is a large family that carries out the same basic chemical reactions. The cytochrome P450 monooxygenases are located in the endoplasmic reticulum, a highly organized and a complex network of intracellular membranes that is particularly abundant in tissues such as the liver. When these tissues are disrupted by homogenization, the endoplasmic reticulum loses its structure and is converted into small vesicular bodies known as microsomes. CYP biotransformations, that is monooxygenation. Chemically diverse small molecules are converted generally to more polar compounds. Reactions include aromatic oxidation hydroxylation, aliphatic hydroxylation, which further includes oxidation of olefins, that is alkenes, oxidation at benzylic carbon, oxidation at allylic carbon, oxidation at aliphatic and alicyclic carbon atoms, oxidation at carbon atom in alpha position to a carbonyl and imines. Next is oxidation involving carbon heteroatoms, that is CN, CO, CS and CX, which further includes oxidative and dealkylation, oxidative deamination, oxidative O-dealkylation as oxidation desulfuration, oxidative dehalogenation. Oxidation of aliphatic and alicyclic carbon atoms. Alkyl or aliphatic carbon centers are subject to mixed function oxidation. Metabolic oxidation at the terminal methyl group often is referred as omega oxidation, and oxidation of the penultimate carbon atom, that is next to the last carbon, is called omega 1 oxidation. The initial alcohol metabolites formed from these enzymatic omega and omega 1 oxidations are susceptible to further oxidation to yield aldehyde, ketones or carboxylic acids. Alternatively, the alcohol metabolites may undergo glucuronide conjugation. Aliphatic omega and omega 1 hydroxylations commonly take place in drug molecules with straight or branched alkyl chains. Numerous barbiturates and oral hypoglycemic sulfonylureas are subjected to omega and omega -1 hydroxylation. Ibuprofen undergoes reaction to give carboxylic acid metabolite and tertiary alcohol metabolites. For example, meprobamate, glutathione, phenylbutazone also undergoes omega -1 oxidation. Oxidation involving carbon heteroatom systems. Nitrogen and oxygen functionalities are commonly found in most drugs and foreign compounds. Sulfur functionalities occur only occasionally. Metabolic oxidation of carbon-nitrogen, carbon-oxygen and carbon-sulfur systems principally involves two basic types of biotransformation processes. A. Hydroxylation of the alpha carbon atom attached directly to the heteroatom, that is nitrogen, oxygen, sulfur. The resulting intermediate is often unstable and decomposes with the cleavage of carbon heteroatom bonds. Oxidative NO and S dealkylation as well as oxidative deamination reactions fall under this mechanistic pathway. B. Hydroxylation or oxidation of the heteroatom, that is NS only, example, and hydroxylation, N-oxide formation, sulfoxide and sulfone formation. Oxidation involving carbon-nitrogen systems. The following discussion divides nitrogen containing compounds into three basic classes. First, aliphatic, primary, secondary and tertiary, and alicyclic, secondary and tertiary amines. Second, aromatic and heterocyclic nitrogen compounds. Third, amide. Metabolic oxidation of tertiary aliphatic amines. The oxidative removal of alkyl groups, particularly methyl groups, from tertiary aliphatic and alicyclic amines is carried out by hepatic cytochrome P450 mixed function oxidases enzymes. This reaction is commonly referred to as oxidative and dealkylation. The initial step involves alpha carbon hydroxylation to form a carbinolamine intermediate, which is unstable and undergoes spontaneous heterolytic cleavage of the CN bond to give a secondary amines and a carbonyl moiety. In general, all alkyl groups such as methyl, ethyl, isopropyl are removed rapidly. N-dealkylation of the T-butyl group is not possible by the carbinolamine pathway because alpha carbon hydroxylation cannot occur. Tertiary amine undergoes a reaction to give carbinolamine intermediate, which is unstable, which undergoes heterolytic cleavage of CN bond to give secondary amine and carbonyl moiety. For example, imipramine undergoes to give a reaction, that is des-methyl imipramine, which further undergoes reaction to give bis-des-methyl imipramine. Metabolic oxidation of secondary and primary aliphatic amines. Secondary amines, either parent compound or metabolites, are susceptible to oxidative N-dealkylation, oxidative deamination and N-oxidation reactions. The primary amine metabolites formed from oxidative dealkylation are susceptible to oxidative deamination. This process is similar to N-dealkylation in that it involves an initial alpha carbon hydroxylation reaction to form a carbinolamine intermediate, which then undergoes subsequent carbon-nitrogen cleavage to the carbonyl metabolite and ammonia. If alpha carbon hydroxylation cannot occur, then oxidative deamination is not possible. Before moving to our next module, that is about oxidation of secondary aliphatic amines, I would like to suggest you to visit our website www.cec.nic.in for further readings and learning of more topics associated with your B.Pharm syllabus. Here you will find many lectures, e-content, FAQs, LORs and many more. Hope this site will be a catalyst of learning for you. And oxidation. Minor metabolites pathway for secondary aliphatic amines. Metabolic N-oxidation of secondary aliphatic and alicyclic amines leads to several N-oxygenated products, and hydroxylation of secondary amines generates the corresponding N-hydroxylamine metabolites. Often, these hydroxylamine products are susceptible to further oxidation, that is either spontaneous or enzymatic, to the corresponding nitrone derivatives. While in case of primary amines, the final metabolic products is an oxime. For example, amphetamine undergoes reaction and hydroxyamphetamine, which further produce imine and oxime. Then oxime further produce phenylacetone. Metabolic oxidation of aromatic amines. The biotransformation of aromatic amines parallel the carbon and nitrogen oxidation reactions since lot aliphatic amines. Tertiary and secondary aromatic amines are encountered rarely in medicinal agents. In contrast, primary aromatic amines are found in many drugs and are often generated from enzymatic reduction of aromatic nitrocompounds, reductive cleavage of azo compounds and hydrolysis of aromatic amides. Tertiary aromatic amines undergoes N-oxidation to give N-oxide, which undergoes carbon hydroxylation to give carbinolamine derivative, which on hydrolysis give aromatic amides. Now dear friends, we have come to our last module, that is module 5. Metabolic oxidation of amides. Amide functionalities are susceptible to oxidative carbon-nitrogen bond cleavage via alpha carbon hydroxylation and N-hydroxylation reactions. Oxidative dealkylation of many N-substituted amide drugs and xenobiotics has been reported. Diazepam undergoes reaction to give an intermediate which on hydrolysis give desmethyl diazepam. Metabolic oxidation of compounds containing CS bond. In case of sulfides, we can observe 3 different metabolic oxidations, that is S-dealkylation, desulfuration, S-oxidation, considered being the major pathway. The first 2 processes involve oxidative carbon-sulfur bond cleavage. S-dealkylation is analogous to O and N dealkylation mechanistically, that is, it involves alpha carbon hydroxylation and has been observed for various sulfur xenobiotics. Here, in this case, we can observe the cleavage of CS bond or S-dealkylation. For example, which in the case of thiopental, there is replacement of S atom with O atom lead to produce phenobarbital. Thioridazine gives ring sulfoxide, which on further reaction give ring sulfone. Well, thioridazine also give mesoridazine and sulforidazine. Dear friends, now coming to our summary section. Phase 1 reaction, also termed non-synthetic reactions, may occur by oxidation, reduction, hydrolysis, cyclization, decyclization and addition of oxygen or removal of hydrogen, carried out by mixed function oxidases, often in the liver. These oxidative reactions typically involve a cytochrome P450 monooxygenases, often abbreviated CYP, NADPH and oxygen. The classes of pharmaceutical drugs that utilizes this method for their metabolism includes phenothiazines, paracetamol and steroids. A common phase 1 oxidation involves conversion of a CH bond to a COH. This reaction sometimes convert a pharmacologically inactive compound, that is a prodrug, to a pharmacologically active one. By the same token, phase 1 can turn a non-toxic molecule into a poisonous one, that is toxification. The following topics have been covered in the present topic: oxidation of aliphatic and alicyclic carbon atoms, oxidation of carbon-nitrogen systems, oxidation of carbon-sulfur systems. With all these information, here we comes to the end of today's lecture, that is phase 1, third. I will be back with few more lectures in this series. Do keep in mind what we discussed today. Time for you to self-study. If you want to learn more and enhance your knowledge, you may log on to our website for MCQs, quiz, LORs at www.cec.nic.in. Till then, keep studying. Goodbye. Thank you.